Hello everyone. I hope you all are doing good. Welcome to your own channel Food Tech Network. As we started our third component of food products, that is enzyme, and already have completed a lecture on it. If you have not watched that, I request you to watch that so that this lecture will become more clearer to you. So in the last lecture we discussed about some of the unique properties of enzyme, their basic classification, how are they nomenclatured, and there are six different classes which is based on the type of reactions they catalyze, their mode of action, the factors which affect the rate of enzyme activity. Today we are going to discuss some further topics which are more important as your career as well as competitive point of view. Today we are going to discuss about the isolation of enzyme, the way by which we isolate enzymes, enzyme immobilization, a process, very famous process, enzymatic browning, and discuss some important industries wherein enzymes are applicable, such as alcohol, dairy, bakery, fruit and vegetable industries. So without wasting time, let us start with it. Isolation of enzymes. See, there are some processes and some concepts in our subject which if you will try to learn or memorize or understand by reading long pages, long number of pages and a lot of data, you will become very much confused. So it is very easy and the best way to make a flowchart out of the long processes to make it more clearer, right? Now, what is isolation of enzymes? When I say I am isolating something, it means I am extracting something. We know that although we can even through biotechnical logical processes, there have been certain advancements where enzymes have been manufactured artificially. But naturally, enzymes are found within the cells of various kind of organisms, because these are biological molecules. They can be found in our body, in the body of humans, in the body of animals, in plants, in the cells of microbes, fungus, and more in all the living beings, because they are the basis of life. Now, how do we isolate these enzymes from such complex sources? So the first step is to identify the source of enzyme. अगर हमें जो एंजाइम चाहिए हमें पता ही नहीं होगा कि वो कहाँ पर प्रेजेंट है, तो हम उसे निकालेंगे कैसे? सो सबसे पहला स्टेप होता है select the source of enzyme, कि एंजाइम मौजूद किधर है. Enzyme, as I said, can be present in an animal, in a plant, in a microbe, anywhere. And the third process, as I said, is the biotechnological process, that is our DNA technique by which we can manufacture it biotechnologically. Now, after identifying the source, we can do these three processes. We can perform these three processes to extract this out. Now, I will give you unique examples to make this more clearer. For example, if you are having an enzyme, present in the cell of an animal or plant or microbe, say we have a particular enzyme, for example, you can say zymase, that is present in the yeast, and we want to extract it out. So where is it present? It is present inside the cell of a microbe, and we need to take it out. So first step will be cell lysis. We need to destroy the components of cell. The cell wall, if present, will be destroyed. In this case, since we have the yeast, it has a fungal cell wall. Then we will remove the cell membrane. The cytoplasm will be removed, and all the components will be removed, and we will get an extract. What is enzyme extract? Enzyme extract is a solution which contains enzyme. It is not at all pure enzyme. So we can perform cell lysis. Now if you want to, if at all possible, you can take the enzyme out at this step also. We have particular filters which can only allow the enzymes to pass through them and will retain all other cell debris. Centrifugation, which is a process based on density, can also be implied, which will remove all other components and retain our enzyme. This can also be implied, and enzyme extract is produced. Now, how to remove other components is a special process. For example, we can do the chemical treatment. We have specific chemicals, say Triton 100X. It is a detergent. It can be used to remove all other components, and enzymes only can be saved, and these can later be filtered out and can be extracted. High pressure treatment can be done to remove other components. Sonication, and that is use of ultrasonic waves, can also be used, and through this method or by removing some other components, we can get a purer form of enzyme called as crude enzyme. But still it is not pure, because some small molecules, for example some nucleic acids, some fat molecules, still can persist over the enzymes, because as I said, enzymes are also protein. They are not found in an isolated manner. They're found in the conjugated manner. A carbohydrate small molecule can be attached to it. It can be attached to a lipid. It can be attached to a nucleic acid. So we need to remove them also. And for that we have some specific processes. A very famous process is salting out. Salting out is a process via which we add a chemical, that is ammonium sulfate, AlSO4, and by addition of ammonium sulfate or aluminium sulfate, we can remove some components which are not enzymic in nature, and enzymes alone will be retained. The nucleus treatment, nucleus itself is an enzyme. It can be used to remove the nucleic acids. DNA, RNA can be removed. So being an enzyme, nucleus can also work. Lipase is also an enzyme. It is used to remove the lipid particles. Fatty acids crystal will be removed from it, and only pure enzyme will be there. So what happens? We have removed the larger components during earlier processes, and by using these three processes of using ammonium sulfate or aluminium sulfate, that is Al2SO4 thrice, and nuclease lipase, we can remove the persisting small molecules also. Now what we get is called as dilute enzyme solution, because enzymes are present here. They are in purer form. We do not have any other component, but still they are dilute. Now we want a concentrated thick mixture of enzyme, and for that we have some again advanced techniques such as ultrafiltration, utilizing a special membrane and other such techniques accompanied with it. Ion exchange chromatography, gel chromatography, affinity chromatography, which will give us completely concentrated enzyme, and this enzyme is packaged and is sent for industrial and other such uses. Now next a very important process is enzyme immobilization. Since we are talking about utilization of enzymes in food industry, so we should know that how do we use enzyme in food industry. We have large tons of containers, a ton capacities container in the food industry, or for that matter of fact any industry. So do we do the same process as it's done in biological system? No, we need to produce products in mass, in large quantities, and for that a technique, that is enzyme immobilization, is utilized. To be technical and scientific, immobilization simply means we restrict the movement of enzyme to a particular range of स्पेस. मतलब अगर मैं साफ शब्दों में बोलूँ तो हम एंजाइम्स को एक पर्टिकुलर जगह पर रोक करके लोकलाइज करके रख देते हैं, जिससे वो इधर उधर ना जा सके, और एक सॉलिड स्ट्रक्चर हमारे पास आ जाता है जिसमें एंजाइम्स एम्बेडेड होते हैं, और जहां पर हमें एंजाइम्स की जरूरत पड़ती है तो इस सॉलिड स्ट्रक्चर को हम वहां पर डालते हैं, एंजाइम्स अपना एक्शन करते हैं और फिर हम इस स्ट्रक्चर को वापस निकाल देते हैं. अब आपको जानना चाहिए कि इसका यूज क्या हो सकता है, क्योंकि हम चाहे तो सिंपल एंजाइम्स भी डाल दें, सिंपली डाल दें एंजाइम्स को, फिर इमोबिलाइज या लोकलाइज करके क्या फायदा होगा. तो इफ वी टॉक अबाउट सम ऑफ द यूनिक बेनिफिट्स, देन इट इज दैट प्रोटेक्शन फ्रॉम डिग्रेडेशन एंड डीएक्टिवेशन. दैट इज बिकॉज if you are simply adding enzyme in the solution, some factors of solution like pH and other such things may degrade the enzyme and deactivate, and our product will not be formed. All the cost will go in vain. The second unique benefit is the reuse of enzymes for many reaction cycle. That is the unique benefit. For example, if you are simply adding a mixture of enzyme in the solution, you cannot take it back. You have, after from the product, you have to again filter it out, and that will increase your cost of input. Rather than if you are using immobilized enzyme, you can simply insert the enzyme stick in the solution and then take it out, and it can be used for multiple times. That will be very cost beneficial. You can stop the reaction at any time. For example, if you are making for example a product that needs a particular concentration and that has been achieved, and now you want to stop the reaction, so immediately you can pick the stick of enzyme up, and that would not have been possible if enzymes would have simply added into the solution. It also enhances the stability. The rate, although get decreased, but the stability definitely increases, and the product is never contaminated with enzyme. Some of the ideal substrate onto which the enzymes is laid down is silicon, bentonite, cellulose, activated charcoal, and other such biomolecules. Some special methods of enzyme immobilization which you just need to know, no need to go into much details, is cross-linking, covalent bonding, entrapment, encapsulation, sorption, which can be physical, chemical, biological, etc. Right. So after understanding two important industrial characteristics of enzymes, that is the enzyme immobilization, which we studied here, and isolation of enzyme, now we are going to talk about a very important phenomena, that is enzymatic pruning. You must have seen that when you keep the cut brinjals, that is eggplant, out, or the apples or potatoes out in the air, then it develops a brown color and the tissues become very soggy kind in nature. Why does this happen? This definitely decreases the sensory attributes of the product. This is because of the action of a particular enzyme which we call PPO, or polyphenol oxidase. Some products are naturally very rich in polyphenolic compounds such as apple, brinjal, potato, many fruits, etc. And they also contain an enzyme naturally that is known as polyphenol oxidase. What it does, it acts on these polyphenolic compounds in the presence of oxygen and copper as a promoter, and convert them into some large polymers which have brown to black color. So the reactant, as I said, for example, you can see this reaction 2,4-dihydroxyphenol is a polyphenol found in apple. In the presence of oxygen, copper as a promoter ion, this polyphenol oxidase converts into orthoquinone and ultimately forms a brown pigment that is a large polymer known as melanin. This is a brown color, and a similar phenomenon is seen in other such products like brinjal and all. Now, how can it be prevented? Because it decreases the palatability of the product. It can be inactivated. One process is that we inactivate the enzyme. How can we inactivate? By blanching. If you don't know, blanching is a process by which we, for a short time, we expose the product to 100 degrees Celsius water or steam, and then we rapidly cool it down. By this method, the enzymes becomes inactivated. So the PPO will not work because it has become denaturated or inactivated. Since the second step which can be used is chelating copper ions using some chelating agents like sorbate, citrate, EDTA. We know that without the presence of copper ion, the enzymes will not start working. So just remove the copper ions by using such unique equipments. We can use antioxidants which will keep oxygen away like ascorbate, tocopherol, TBHQ, BHT, etc. And the last and the most simple thing is maintaining anaerobic condition. अगर ऑक्सीजन होगी ही नहीं तो एंजाइमेटिक ब्राउनिंग कभी हो ही नहीं सकती. Right. So after understanding these unique processes, let us begin with industrial considerations. Beginning with alcohol industry. Alcohol industry is a very large industry all over the world and also in India. And alcohol production is not a latest process. It has been done since thousands of years. Even in our old historic textbooks, we have such things. The production of alcohol, what we call as som in Sanskrit. Correct. Enzymes play a very vital role in alcohol and brewing industry. They are utilized in the production of various types of alcoholic beverages like wine, beer, fanny, whiskey, rum, sake, and whatnot. Alcohol production comprises of breaking down complex polysaccharides into simpler sugars. That is the first step. The complex polysaccharides, say starch or cellulose, are to be broken down into simple sugars, and the enzymes present in yeast act on this simple sugar to produce alcohol or ethanol. Right. Now let us see the biochemical aspect of alcohol production. The first process which I said is to break down the complex polymeric carbohydrates into simpler ones. That is generally done by using some enzymes. For example, alpha 1,4 amylase. Now alpha 1,4 amylase is a particular enzyme which breaks down the amylose part, or which breaks down the glycosidic bond between adjacent glucose molecule present in starch and release maltose. Now what happens if you are using this enzyme? The starch molecule, that is a complex carbohydrate, will be broken down into disaccharide molecules, that is maltose. And after getting maltose, as is composed by two molecules of glucose, is further broken down by enzyme maltase. So the second enzyme is maltase, which will form glucose, and at last zymase, which is the main enzyme, will convert this glucose molecule so produced into ethanol and carbon dioxide. You can see starch molecule by amylolytic enzymes is converted into glucose, wherein we also use maltase. The glucose is converted into ethanol and carbon dioxide by using yeast. Now, this is the process which is common in making beer, wine, and all the alcoholic products. But in the beer industry, enzymes have another role to play. What is that? What happens when beer is subjected to temperatures lower than 0 degrees Celsius? We know that people prefer a chilled beer. Even in the shops, you must have seen chilled beer, या ठंडी beer available. So, what happens by subjecting beer to chill temperature? It develops a turbidity or a haziness which is not appealing to the customers. People think that no, the beer is not pure, it is of cheaper quality, but that is not the case. What happens at lower temperature, some compounds like flavonoids reacts with the proteins present in the beer, and they form some complex molecules or colloidal particles which induce this turbidity in cloudy structure. This can be definitely removed by using enzymes, a specific class of enzymes known as protease, and the most highly utilized enzyme is papain. We derive it from papaya latex. What it does, it breaks these hazing compounds. The bond between flavonoids and proteins is broken down, and such colloidal particles break up, and the chill hazing is prevented. In industry or in commercial parameters, papain 20% solution is sold in by the name of chill proof, because it makes beer chill proof by not letting the haziness induce. Correct. Coming to the bakery industry and alcohol industry both utilizes enzymes for similar structure. The mechanism is same. We need to convert the complex molecules of carbohydrate into simpler ones, which are broken down into glucose, and ethanol and carbon dioxide is produced. But alcohol industry is concerned with only ethanol, and the bakery industry is concerned with the carbon dioxide, because carbon dioxide has a leavening property. It leavens the baked products, it leavens the dough and make it very fluffy and porous, which the consumers demand. The raw material for all bakery products in the bakery industry chiefly includes flour, atta, whole or refined. Enzymes to a large extent impacts the baking property of flour and dough prepared from it. Various attributes like elasticity, leavening, flavor, taste, etc. are function of enzyme. So as I said, leavening, which is means alcoholic fermenting enzymes are used for leavening, but some other properties like elasticity, flavor and taste are also impacted by enzyme. Let us see how. The enzyme amylase acts on amylose and makes convert it into maltose. The baker C's convert the maltose into maltase, maltase to glucose, glucose to ethanol and carbon dioxide, which helps in leavening. So this is what I already told you. Now what happens? A scaling phenomenon is observed in the baked products. I will suggest you to do this at your home also. Just take a packet of bread, eat as much as you want. Just keep one bread separate. Put this bread in the refrigerator and wait for 2 to 3 days. What you will observe that after 2 to 3 days, the bread has become very hard. Why is it this so? This is because at lower temperature, even by keeping for a longer period of time, the starch molecules present in the bread, they start rearrangement. They start rearranging themselves, and a very tough structure is formed. This process is known as staling. How can this be prevented? This can be prevented by using this same enzyme amylase, which breaks them down into smaller fragments, and staling is prevented. The third most important feature of baked products is there is elasticity and stretchability, कि उनको हम कितना खींच सकते हैं, चबाने में उनको कितनी आसानी है. This can be controlled by protease. We know that elastic properties of bread is due to enzyme, a component that is gluten, protein. So protease acts upon these gluten fibers, and such a reaction takes place that the elasticity and stretchability is controlled and monitorized. Even for making white, we have two kinds of bread in the market: white bread and brown bread. Some people prefer white bread. Now how does the bread attain this color? This is because we oxidize the pigment present in bread. The pigment is oxidized by using enzyme lipoxidase, and a white colored bread is produced. So this is the process of leavening, amylase, maltase, enzymes, which I have already told you. Also, I will like to discuss with you a unique product, that is gluten-free flour. See what happens. Some people have allergy with gluten. They cannot digest it. They start getting allergic reactions into their body by consumption or intake of gluten. So just to avoid this, what people do, Ashirwad, they're one of the very famous brand in the field of flour, part of ITC, has introduced gluten-free flour. What it does, it uses an enzyme named as DX11. It helps in removing the gluten. It hydrolyzes the gluten, and gluten is not present in its allergenic form, and just to give that sticky nature, because you may wonder अगर gluten होगा ही नहीं तो फिर आटा गूंदेंगे कैसे. So this can be done by using some additives, some emulsifiers such as xanthan gum, guar gum, which helps in attaining a specific dough-like structure. Now coming to the next industry, that is vegetable and fruit industry. See, even if we do not talk about processing, if I talk about nature also, enzymes play a very important role in getting the crop produced. How? The ripened fruits and vegetables have specific color, flavor, odor, taste due to various enzymatic processes. Enzymes like polygalacturonase, pectin esterase, cellulase, amylase, etc. breaks down complex polysaccharide like pectin, cellulose, starch and convert them into simple units. These simple units are modified into acids which may form some colorful compounds, flavor compounds, etc. These enzymatic activities make fruits more firmer. In ripe, when unripe, when raw, they are very hard. आपने देखा वो कच्चे होते हैं तो बहुत hard होते हैं. But by the enzymatic action, they become very pulpy, very soft. The TSS or the total soluble solid content is increased. Flavor, taste, and odor is developed in the pulp, which is very much appealing. Other than this, some enzyme like DX1, TSSI help in carotenoid production, which gives a specific bright color to the fruit. Thus, enzymes develop the peculiar attribute into the fruits and vegetables, making them develop specific color, firmness, texture, flavor, and taste. These activities of enzymes doesn't stop in some commodities like climacteric fruits. You must have seen, अगर आप घर पे आम को ला के रख देते तो आम पकता रहता है. अगर आप घर पे फॉर एग्जांपल किसी और फल का मैं एग्जांपल दूं, जैसे कि आप देखें कि नींबू को लेकर के आओ आप, या फिर आप अंगूर को लेकर के आओ तो पकता नहीं है, जैसा आप उसको लाते हो वो वैसे ही रहता है. गर्मी के कारण वो गल जरूर सकता है लेकिन वो पकता नहीं है. लेकिन आम हो गया, और 2-3 और फ्रूट्स हो गए, अगर आप नोटिस करेंगे वो पकते हैं. केले भी हो गए, आप उसको हरा लेकर के आते हो तो कुछ समय के बाद पक जाता है. तो जो फ्रूट्स के अंदर एंजाइमेटिक एक्टिविटी चलती रहती है उनको हम लोग बोलते हैं क्लाइमेक्टेरिक फ्रूट्स. एथिलीन प्रोडक्शन के बाद एंजाइम्स इनमें वर्क करते रहते हैं इवन आफ्टर टेकिंग देम आउट ऑफ द प्लांट बॉडी, लेकिन नॉन क्लाइमेक्टेरिक में ये नहीं दिखता. क्लैरिफाइड जूस एंड बेवरेजेस, आपने नोटिस किया होगा आप घर पे कोई जूस निकालते हो तो उसमें ना फाइबर्स आते हैं, रेशे आते हैं जो कि मुंह में फंसते हैं. लेकिन जो हमारे पास बड़ी बड़ी इंडस्ट्रीज के जूस है, बी नेचुरल, रियल, इनके जूस एकदम क्लियर होते हैं. व्हाई इज दिस सो? बिकॉज वो पेक्टिनेस और सेलुलेज जैसे एंजाइम यूज करते हैं जो कि इनकी फाइबर्स को ब्रेक डाउन करते हैं सिंपलर यूनिट्स में, और क्लियर जूस ऑब्टेन होता है. एक और इम्पॉर्टेंट चीज आप नोटिस करते होंगे कि कई बार क्या होता है, जो कैंड वेजिटेबल्स या फ्रूट्स होते हैं, अगर आप उनको कैन के थ्रू रखो या उनको ग्रीन हाईली एसिडिक कंडीशन में रखो तो वो ऑलिव कलर के हो जाते हैं. जैसे आप ये देखो, ये इनका कलर ऑलिव हो जाता है. तो इस चीज को प्रिवेंट करने के लिए भी एंजाइम्स यूज होते हैं. एक्चुअली हमें पता है ग्रीन कलर सिर्फ और सिर्फ क्लोरोफिल के कारण होता है, मतलब मेनली क्लोरोफिल के कारण होता है. यानी ग्रीन कलर क्लोरोफिल का तब हटता है जबकि हमारी एसिडिक कंडीशन में ये सेंट्रल मैग्नीशियम आयन क्लोरोफिल से हट करके फियोफाइटिन बनाए, एंड इसके केस में इसका कलर ऑलिव सा हो जाता है, डल ग्रीन हो जाता है. इसको प्रिवेंट करने के लिए हमारे पास एक एंजाइम होता है क्लोरोफिलेज, जो कि इस क्लोरोफिल मॉलिक्यूल से फाइटोलिन को रिमूव करता है, और हमारे पास एक पर्टिकुलर क्लोरोफिलाइट कंपाउंड बनता है जो कि ब्राइट ग्रीन कलर का होता है. मतलब अगर आप मैग्नीशियम चीलेटेज एंजाइम का यूज करेंगे तो ये डल ग्रीन बन जाएगा, लेकिन अगर आपको ग्रीन कलर चाहिए जो कि अपीलिंग हो तो आप क्लोरोफिलेज यूज करोगे जिससे ब्राइट ग्रीन कलर रहे. सो दिस वॉज ऑल अबाउट आवर वीडियो. मुझे लगता है कि आपको सारी चीजें समझ आ गई होंगी. फिर भी कोई डाउट हो तो यू कैन ड्रॉप योर डाउट्स डाउन इनटू द कमेंट सेक्शन, और इवन पुट योर डाउट्स in our website and the contact numbers given. And today we discussed basic things like enzyme immobilization, enzymatic browning, how do we extract the enzymes out. We took over to various industries like alcohol industry, bakery industry, vegetable and fruit industry, etc. And in the next sections we will discuss some more important industry. So keep learning and keep enjoying. Thank you.